It's time to start. So, welcome everyone, and it's my pleasure today to introduce Jason Morrow. And Jason is finishing up his master's degree here in soil science. Jason got his Bachelor of Science at the University of Wyoming in civil engineering, and actually did so well that he was part of he was inducted into the National Honor Society of Engineers at that time. And later on, he got the bug for agriculture when he went and worked with the Peace Corps in Zambia, and was a water resource engineer with them. And clearly, at that point, he was working with a lot of subsistence farmers at that time, and he could see the link between community health and soil health. And he started his research here almost a year and a half ago or so, funded through the REACH project, so Regional Approaches to Climate Change, and his topic is soil health. And so, Jason, we'll turn it over to you for now, and I'll remind everyone to keep track of their evaluations and turn those at the end. Thank you.
Well, thank you, Dave. Thank you, everyone, for being here today. So, my topic of the discussion today is my master's research on tillage, cropping intensity, and climate effects on soil, carbon, and nitrogen within a soil health perspective. And as Dave said, this research has been funded by REACH, Regional Approaches to Climate Change. So, an outline of my talk today. First, I'll do a brief introduction on soil, carbon, and nitrogen and its importance to soil health. I'll briefly go over my soil analysis that I've performed. And then I'll address 4 research questions, which are listed here and which I will show as I come to them. And then lastly, conclusions. So, first of all, what is soil health? It's been discussed as far back as the 1940s in terms of soil organic matter and the impacts of climate and management on soil organic matter. There are several definitions of soil health that have been presented in the literature. However, a uniting theme of these definitions is the ability of the soil to function effectively at present and into the future without jeopardizing environmental health. In the context of agricultural systems, this means maintaining productivity and efficient utilization of resources while continuing to enhance the essential services provided by soils, which, as most of us have learned by this point, include the promotion of plant growth, cycling of nutrients, provision of habitat for soil organisms, and its crucial role in the water cycle. Furthermore, as soil organic matter is crucial to these functions, it's soil organic matter that is often associated with soil health. In further understanding soil organic matter, it's often divided into various pools based on turnover time. In its simplest form, it can be represented into 2 pools, both an active pool and a passive pool. These pools are based on turnover time, with the active pool turning over from anywhere from a few days to several years, and the passive pool with the turnover time hundreds of years or more. The active pool is commonly associated with providing energy for the soil food web, its role in soil aggregate structure, micronutrient chelation, and nutrient mineralization, whereas the passive pool is more commonly associated with cation exchange capacity, water holding capacity, and soil structure. It's this active fraction which is commonly targeted when making evaluations of soil health and management impacts of soil health. Additionally, soil organic matter is not homogeneous. It's comprised of various components ranging from fresh plant residue to more inert humic substances. However, it is generally accepted that it's comprised of approximately 58% carbon, with nitrogen variable. So it's the active carbon and nitrogen which is typically targeted in assessing soil health and management impacts on soil health. Furthermore, as it's important to assess the overall soil profile when assessing total carbon and nitrogen stocks. In evaluating soil health, it's the upper soil profile which is commonly the focus of attention, as it exerts such a strong influence on important factors that influence soil health, including erosion potential, water infiltration, and nutrient cycle and conservation. And it's the upper profile, the upper 10 centimeters, which has been the focus of my study. There are many analyses which can be used to capture carbon and nitrogen properties within soil. The ones that I have utilized, and which I will be presenting findings today, are presented here and include permanganate oxidizable carbon, also called POC, microbial biomass carbon and nitrogen, water extractable carbon and nitrogen, potential carbon and nitrogen mineralization, and acid hydrolyzable carbon and nitrogen, which actually divides carbon and nitrogen pools into two pools based on the hydrolyzable and non-hydrolyzable fraction, or the active and the passive fraction. And then total soil organic carbon and nitrogen. This is a depiction of the study area. It spans 4 agroecological classes, which are distinct growing zones, and includes 5 sites. These sites are Cambridge Farm, just outside of Genesee, Idaho, PCFS, just outside of Pullman. These are in annual cropping zones. Pendleton, Oregon, which is in an annual crop transition zone. And Morrow, Oregon, a grain fallow zone. And then lastly, Prosser, which is an irrigated site. As you can see, I've depicted here, these sites span a range of precipitation, mean annual precipitation and mean annual temperature gradients. The wettest site being Cambridge at 27 inches, down to Prosser, the driest site at 8 inches, and then mean annual temperature inversely related to this gradient, with 47 degrees at Cambridge up to 51 degrees average mean annual temperature at Prosser. So these sites were selected not only for this climatic gradient that they represent, but also for the crop rotations present at each site, which include both tillage comparisons and cropping intensification comparisons. Here I have listed the tillage comparisons, which are present at 4 sites. At Cambridge, with 14 years of management history with these rotations, includes winter wheat, spring cereal, and a spring legume rotation. Pendleton has 17 years of management history with the winter wheat fallow rotation. These are both till and no-till. Morrow, with 10 years of management history, is a winter wheat fallow rotation. And then Prosser, again, the irrigated site is the most recent site, and has a winter wheat corn potato rotation. And then the cropping intensification comparisons are present at 3 of the sites. PCFS, with 13 years of management history, has 2 perennial rotations, an organic rotation, which includes alfalfa, spring cereals, and a spring legume, and then a winter wheat spring re-rotation with either spring barley or spring legume. And then Pendleton has intensification represented by winter pea inserted in rotation, and this was only recently done, so it only has 1 season of history. And then Morrow, with 10 years of history, has annual cropping with winter pea, cropping 2 out of 3 years with winter wheat and spring barley, and then winter wheat fallow cropping. In order to perform my analysis, I found it useful to quantify these treatments. Typically, in capturing cropping intensification, it's common to utilize biomass production. However, this information is not consistently available across all sites for all rotations. So I found it necessary to capture it in a different way. So up top here represents cropping intensification values, which I devised based on the duration from planting to harvesting, or for perennials from the approximate time annually when they're actively growing. So .83 represents 10 months out of the year for perennials. .75 represents 9 months out of the year for fall planted crops such as winter wheat. And .33 represents 4 months out of the year for spring planted crops such as spring wheat, spring barley, garbanzo beans. Then, in order to quantify tillage, I utilized a system developed by the NRCS called the Soil Tillage Intensity Rating. This is commonly used in the Russell equation and represents a relative degree of disturbance represented by each operation for each crop in rotation. For example, here I show spraying herbicide, which is relatively low disturbance, has a stir rating of 0.15. More disturbing, of course, cultivation with a chisel plow, 52.6. Moldboard plow, even more disturbing, 65. So each management operation for each crop in rotation is assigned a value, and these are summed across rotation and divided by the number of years in rotation to come up with an annualized stir rating, which is representative of the tillage. And the cropping intensification is also an annualized value. So in doing that, this is a representation of the result by site. You can see here the upper group, higher stir rating. These are the rotations that are considered conventionally tilled. In the lower group here on the bottom with the green arrow, these are the cropping intensifications. So when I talk about tillage comparisons, I'm talking about moving along the brown arrow here vertically. So comparing tillage rotations at the same cropping intensification. And then moving horizontally across the green arrow are cropping intensification comparisons at similar stir ratings. Just briefly, soil sampling. Samples were collected this past summer in June and July. And samples were collected from the winter wheat portion of rotations, except for the organic rotation at PCFS, in which spring P was present. As I mentioned, samples were collected to a depth of 10 centimeters with 60 subsamples collected per rotation in bulk. In performing statistical analysis, plots are randomized complete block designs. I utilized ANOVA, PROC-GLM with a p-value less than 0.1, and multivariate stepwise analysis, which I will be presenting later. I used PROC-GRED with model entry and significance at 0.1. And just a quick overview on the soil analysis I performed. Some of the concepts behind them. Total soil organic carbon and nitrogen, pretty straightforward. It just represents the entire soil carbon and nitrogen pool. I utilized the dry combustion method. For acid hydrolysis, in this procedure, a small quantity of soil is ground to a powder and digested with a strong acid solution. What remains after this digestion period is considered the non-hydrolyzable carbon and nitrogen. And this is considered representative of the more passive pools. Whereas the active pool is the carbon and nitrogen that's hydrolyzed, which is the difference between the total nitrogen and soil carbon pool and the non-hydrolyzable fraction, which would represent more of the active pool. Permanganate oxidizable carbon, POC. This is an important one which I'll be discussing quite a bit later, so remember this one. It utilizes a weak potassium permanganate solution as an oxidizing agent in which manganese is reduced in accordance with the amount of active carbon in the soil. And this reduction in color can be captured by spectrophotometry. This particular concentration was developed to attempt to specifically target the fraction of the soil carbon pool that's most influenced by management practices. Additionally, while I performed my analysis in the lab, there's a field kit version making this more accessible to land managers. Microbial biomass, carbon and nitrogen, just as the name implies, this captures the microbial biomass at the time of analysis after a short incubation period at room temperature and water content, fluid capacity. And then water extractable carbon and nitrogen, again, as the name implies, it's the fraction of the carbon and nitrogen pool that can be extracted with deionized water, and is often considered to be the most labile carbon and nitrogen pool, and composed largely of carbohydrates and proteins. Potential carbon mineralization. This is a soil property that is commonly associated with soil health, as it is influenced by soil physical, chemical, and biological properties, and is a measure of soil microbial activity. In performing this analysis, the soils were dried and re-wetted, representing the natural process in the field of drying and re-wetting. And CO2 was measured over a period of time, starting from 24 hours on up to 24 days. And this additionally, just like POC-C, there's a commercialized version of this available, making it more readily measured just for the 24-hour carbon mineralization value. And then lastly, potential nitrogen mineralization. Also an important measure of soil health, as it represents the ability of microorganisms to decompose soil organic matter, as well as provide an important source of soil fertility plant nutrients. And I performed a 28-day anaerobic incubation for this. So going into my first research question, I was looking at across the research area, what was the influence of 60-year mean annual temperature and precipitation on soil carbon and nitrogen levels? So in this region, it's commonly understood that rainfall and subsequent soil moisture drives biomass production and the carbon and nitrogen inputs to the soil, while the temperature is driving decomposition rates. This represents soil organic carbon and its relationship to mean annual temperature and precipitation across the area. As you can see, as temperature increases and mean annual precipitation decreases, soil organic carbon levels decrease. This spans the 5 sites, as I said, and it provides an opportunity to understand the sensitivity of soil organic levels, soil organic carbon levels, soil organic matter to potential climate change. For example, based on average values across the sites, and the slope of the line on the left there, you can see that a 1 degree Celsius change in temperature is associated with a change in soil organic carbon level of 8.3 milligrams per gram of soil. To put this in context, this is approximately equivalent of the shift in soil organic carbon from PCFS to Pendleton. Not necessarily catastrophic, but potentially influencing some important soil functions. In further looking at the influence of climate on the soil properties I measured, I performed a multivariate stepwise analysis with temperature, mean annual temperature, mean annual rainfall, along with management factors, being cropping intensity, the stir rating, as well as pH, to understand how these potentially explanatory variables are interacting to explain the observed variation in the soil properties. As you can see here, represented by what I have circled there, temperature is an overriding factor in dictating the soil properties that I've measured, explaining as much as 71% of the variation across sites in soil organic carbon total nitrogen as much as 78%. However, tillage mostly is not without its impact. You can see down there I've circled the water extractable portion of carbon. The primary management factor explaining its variation was tillage. So this speaks to the idea that in a given area, it's the climate that's dictating soil organic matter saturation levels. However, within these limits established by the climate, management practices are not without their influence. So, in light of this, I looked into the effects of tillage and cropping intensity on soil health as measured by soil carbon and nitrogen measurements. As you can see, indicated by these statements by researchers, different individuals have had different levels of success in capturing management influences on soil quality. So next, I'm going to be presenting a series of slides that depict the relationship between POC and soil organic carbon and nitrogen before I go to specific findings on tillage and cropping intensity. As you can see here, POC exhibits a strong relationship with soil organic carbon and nitrogen, indicating that it's capturing much of the same information as these two soil properties. So it can serve as a good proxy for soil organic carbon and nitrogen. And then again, the relationship between POC and the non-hydrolyzable fraction of carbon and nitrogen, which, as you recall, represents the more recalcitrant pool, the passive carbon and nitrogen pool, also exhibiting a fairly strong relationship. And again, POC with a hydrolyzable fraction of carbon and nitrogen, also exhibiting a strong relationship, indicating that it's providing similar information. However, when this is compared, POC is compared to the 24-hour carbon mineralization value, this strong relationship is not there. The 24-hour carbon mineralization is representative of more of a highly active fraction of carbon and nitrogen, as well as microbial activity. And therefore, these two parameters, soil properties, are providing different information about the soil. So in looking at these average values across the sites and across rotations, this is the result. And as you can see, I have them listed here by site. They fall into different groups, and this is indicative of the influence of climate, which I've recently talked about. However, management does play a role in where a particular soil falls on here. So the y-axis is associated with the potential mineralization ability of the soil organic matter, the ability of organic nutrients to be mineralized into inorganic nutrients. The x-axis, POC, which, as I just demonstrated, captures the overall soil organic matter pool and can be more associated with stabilization. And then a combination of these two, which is kind of a general trend across the sites, would be increasing mineralization potential while also stabilization, which might be considered ideal. So the idea is to understand how different management practices move a particular soil around on the spectrum with a particular management goal in mind. So in looking at this closer with regards to tillage and cropping intensity on a site-by-site basis, these are results from Cambridge, where these are tillage comparisons, where tillage has resulted in significantly more POC representative of stabilization than till, so that's good. That's what we hope for. And then mineralization. No-till versus till has not had a significant effect on mineralization acreage. There is some noise in the data, in the 24-hour carbon mineralization data, making it a little bit more difficult to detect differences. And then Pendleton, again, no-till, significantly more stabilization associated with no-till than till, as well as with mineralization. Pendleton has 17 years of management, and you can see that no-till has resulted in greater mineralization potential associated with the soil cover. And then at Morrow, again, no-till, greater stabilization than till, and no significant differences detected in the mineralization. And then Prosser, with only 2 years of history, no significant differences in either stabilization or mineralization. And then looking at it on a cropping intensification basis, this is PCFS, which, as you recall, has 2 perennial rotations, an organic rotation, and then two annual rotations with spring barley or spring wheat, along with winter wheat, spring wheat. No significant differences there in stabilization or mineralization. Again, with some noise in the data, making it somewhat difficult to detect differences there. And then Pendleton, no cropping intensification there with winter pea, which has only been in rotation for 1 year. No significant differences detectable there yet, which would be surprising after 1 year. And then Morrow, winter pea, which has been represented an intensification there for 10 years, has resulted in significantly more stabilization than just winter wheat, no-till fallow. These are all within no-till cropping systems. However, mineralization, no detectable significant differences there in mineralization. So these are, here I'm talking about tillage and cropping intensity as management variables. However, this same concept could be applied to different management practices, such as cover cropping, soil amendments, and how they move the soil around on here on this spectrum with a particular management goal in mind. For example, for an organic system, it may be desirable to increase mineralization. What practices are effective at doing that? So this represents the significance of combining these two soil properties and the information that can potentially provide about the soil. So obviously there are several soil properties which can reflect different aspects of soil health. And oftentimes these soil properties can be combined into soil health indices to tell us more about the soil. Next, I will be presenting results from an evaluation of a soil health index, which is being discussed by growers and various entities in the area. First of all, just a brief note on soil health indices. Of course, they should reflect management goals and be sensitive to management practices. They should be sensitive to both climate and soil type. And again, it's important to integrate different aspects of the soil, both physical, chemical, and biological indicators. Ideally, they capture a few key soil properties that are representative of greater biogeochemical processes. Examples that have been successfully used include the Cornell Soil Health Test and the Soil Management Assessment Framework. Here is an index developed by soil scientists out of Texas with the USDA that's been successfully applied in other areas. It utilizes the 24-hour carbon mineralization, which, as you recall, integrates physical, chemical, and biological indicators, as well as the water extractable carbon and nitrogen values from the soil, which is an important source of, has been shown to be an important source of energy for microbial activity for the initial period of microbial activity. Additionally, in general, the soil carbon to nitrogen ratio is often an important indicator of nutrient mineralization. The general rule of thumb that greater than 20-to-1 net immobilization occurs and less than 21 net mineralization will occur. So this index captures the water extractable carbon to nitrogen ratio and its influence on nutrient mineralization. And additionally, in the right to the water extractable carbon and nitrogen parameters there to the right captures just the overall importance of building soil carbon and nitrogen. And the 100 to 10 is representative of an idealized 10-to-1 ratio, reflecting a theoretical greater microbial use efficiency of this water extractable portion of the carbon and nitrogen pool. And these are the results across the 5 sites of the Soil Health Index. And no significant differences were detectable across the sites. And in looking at this closer, I performed a multivariate stepwise analysis with the Soil Health Index as the dependent variable, along with management practices and climate variables. And none of these variables were able to explain any of the variation in the Soil Health Index. In looking at it closer, the individuals who developed the index noted a strong relationship between the water extractable carbon and nitrogen ratio and the 24-hour CO2 mineralization. These are results from the research plots across the sites. And as you can see, there's a handful of sites with higher CDN ratios, but that still had mid-range 24-hour carbon mineralization values. So not necessarily a high degree of sensitivity is evident here. And next, those who developed the index also noted a strong relationship between the water extractable carbon and nitrogen pools individually and the 24-hour carbon mineralization values. As I have indicated here, the C-min 0 to 1, that's the 24-hour value. And the relationship with both the carbon and nitrogen pools is not significant. And then further on, there is significance that shows up later on in this carbon mineralization at 3 days, with the highest being at 10 days. However, as they noted, at 1 day, the significant relationship is not there. So I believe what's needed in light of the fact that the expected relationships were not there within this data set, that the index should be reevaluated with a different data set across the sites or other sites. Also, the concept is sound. Perhaps it's possible to integrate other easily measured soil properties into an index that's appropriate for the area. Next, I will be going into my last research question, which is looking at how the soil carbon and nitrogen measures across the study area relate with nitrogen mineralization. So just briefly on potential nitrogen mineralization, as I've stated previously, it is an important representation of soil health, as it represents the ability of microorganisms to decompose soil organic matter, as well as provide an important source of soil fertility. Additionally, it's an important consideration as it influences nitrogen use efficiency and considering it helps minimize environmental loss of nitrogen. Commonly, total potential soil nitrogen mineralization is determined from long-term incubations, upwards of 210 days or so. Short-term incubations, however, have also been shown to correlate well with these longer-term incubations. However, these shorter-term incubations are still not set up for wide adoption, as 7 days is just prohibited. So there's a need for shorter-term, routine, and accurate methods for predicting nitrogen mineralization, both for improving monitoring soil health, this important soil process. No single nitrogen availability index has shown promise for wide acceptance. So there's a need for understanding on a more regional level what soil properties play into nitrogen mineralization. So in looking at this, first, these are Pearson correlation values of the soil carbon and nitrogen properties that I've measured, along with nitrogen mineralization. For the carbon mineralization values, they were significant but low correlation. I believe this potentially may be because the nitrogen mineralization was an anaerobic incubation, whereas the carbon mineralization was aerobic incubation. So that may potentially explain the low correlation there. However, soil organic carbon, total nitrogen, and epoxy all exhibited a strong correlation as I've shown earlier, these soil properties are related. So if one correlates, we expect the rest to do, of course. C-to-N ratios, not significantly influencing nitrogen mineralization. The C-to-N ratios for this study all fell within a narrow band, typically less than 20 to 1, so not a limiting factor. And then stir, intensity, pH, temperature, and precipitation all were significant. In looking at this closer, again performing a multivariate analysis of N mineralization as the dependent variable and various combinations of management factors, soil carbon and nitrogen properties, and climate factors as potential explanatory variables. The model up top here, including all three management, soil properties, and climate factors, selected soil organic carbon, epoxy, stir, and intensity as the most significant. While epoxy and soil organic carbon are highly correlated, I left them in there as removing one, nothing else, replaced it in the model. And they were able to explain 81% of the variation in nitrogen mineralization across the study area. And looking at just a model of climate factors and management factors, temperature and the stir rating, the tillage intensity, were able to explain 61% of the variation in mineralization across the study area. And then in looking at a reduced model with just easily measured soil properties, this model with epoxy, cropping intensity, and the stir intensity interaction explaining only, again, 61% of the variation. So no better than just consideration of temperature and tillage intensity. So this represents the power of combining multiple properties, easily measured properties in determining N mineralization, and that being an important indicator of soil health, it is possible to understand N mineralization variability with relatively easily measured and determined factors. So, in conclusion, Climate was the dominant factor in driving soil carbon and nitrogen pools. POC is an easily measured and a strong indicator of multiple soil properties, including soil organic carbon, total nitrogen, as well as the hydrolyzable and non-hydrolyzable fractions. And that POC and carbon mineralization considered together capture important information about the soil organic matter pool, both in regards to stabilization and mineralization. Looking at tillage across the REACH study area has had a measurable impact on soil health with no-till, greater than till, in 3 out of 4 sites. And again, tillage, highly active carbon and microbial activity as measured by the 24-hour carbon mineralization was only significantly different at Pendleton, no-till versus till. Cropping intensification differences were within no-till were less apparent. However, at the Morrow site, with winter pea in rotation, Morrow site being a grain fallow region, a drier region, cropping intensification has resulted in significantly greater POC. However, no significant differences were detected in the 24-hour C-min values within cropping intensification comparisons. Soil health index, the initial evaluation was not able to differentiate between management influences as well as show sensitivity to temperature or precipitation. And I believe it should be evaluated at other sites with other data sets, making a determination if it has the needed sensitivity within this area. And that perhaps other soil properties could be combined based on the similar concept. And then lastly, 28-day anaerobic nitrogen mineralization is an important indicator of soil health. And combining multiple factors, easily measured factors, can be a good predictor of N mineralization. With soil organic carbon, POC, tillage and cropping intensity explaining 81% of the observed variation across the study area. And lastly, I would like to thank my major advisor, Dr. Dave Huggins, along with my committee, Dr. Lynn Carpenter-Boggs and Dr. John Reginald, as well as to Dr. Jeff Smith, who was very gracious in allowing me to use a lot of his laboratory equipment performing these analysis, along with many others who have been helpful both either with questions or with helping assisting with laboratory procedures, and of course lab and office needs. Thank you.
Jason, are there any questions?
Yes.
Do you know the depth to which all your tillage practices were done?
I know some of them were about 10 centimeters. I'm not sure about all of them. Yeah.
Was all your sampling in the top 10?
Yeah, top. I focused on the top 10 centimeters.
Some of the tillage might have been further than that.
In a few instances, perhaps. Yes.
What impact was there as far as the amount of residue in each one of those sites on that base?
On the surface?
What difference did the yield make? The amount of residue left over from
What different? So, in looking at the correlation between yield and these,
the different tillage practices or
I didn't specifically look at. I started looking at residue values, and they weren't available across all sites. I did notice that for certain fractions, particularly the non-hydrolyzable fractions, had higher values in no-till versus till, and I think that was probably likely a result of the palm fraction being greater in the no-till sites, the particular organic matter fraction. So that was one particular influence of it.
But there was no tie-in between the yield and tillage.
No, I haven't looked at yield. I agree that would be a good thing to look at. Yes.
So in terms of like when you were looking at precipitation as a factor, did you do something different for crossers since it's irrigated?
No, I just considered the 60-year mean annual value, and it fell, even though it's irrigated, it fell within the trend of the other sites that are not irrigated. Yes, Jessica.
So are these soil health indices being developed primarily for research use or for land management use?
For land management use, for evaluating. So,
Has there been any attempt to sort of incorporate the potential, sort of the potential highs into these values to sort of scale them by climate? In terms of, it's going to be very different from a farmer in Crosser or something looking at these indices versus someone over here in Moscow. And is there a attempt to sort of scale to say, hey, you're sort of meeting 80% of the potential?
yeah, I can't speak specifically to that, but I think the idea is to look at the value and within a specific area and consider it in that context, not necessarily across a broad area, but more of a localized area.
I'm just curious sort of whether they would have something to compare it to. So you say I got a
Right. Well, I know, like for the
0.5.
index that I've showed here, values can range anywhere from very low up to 50. So values that I showed were anywhere from 3 to 8. So, yes.
On your winter rotation, was that taken off as a crop or was that turned down as a winter?
Turned in. Yep. Turned into. Yes.
So why do you think the water extractable carbon isn't working as perfectly as it seems to in Texas or the samples they are using?
Yeah, that's a good question. There is some. Well, the 24-hour carbon mineralization value is variable, so it can make it difficult to identify correlations. So, in performing that, a rigorous statistical analysis, yeah, those properties are just highly variable, so it's hard to pull out that. So, an improved index may be properties that are less variable or easily measured. Yes?
You just noticed on your water check, carbon water check matches in the 24-hour mineralization slide, the graph. It looks like that divergent trends there. Did you look for any distinct differences between those groups? They just went off like roughly separately different.
Yeah, I think so. That was the.
I'm just curious if you looked at that by if there's any distinguishing factor between those two different groups.
So this one?
Yeah.
So these ones here are the higher C-to-N ratios. So you're talking about
Well, if you just broke this up either by size or by other factors, or anything common.
Got you. I have, and Off the top of my head, I'm not recalling. I believe they were associated with CAMH, but I'd have to double check that.
Because if you took some of those out for
Right,
one reason, because there seems to be others.
right, yeah, no, that makes sense.
Got a trend there.
Right.
It's just simple comparison.
Yes.
And we have, I assume these soils were quite different across this area, where they were starting from, whether they're loams, silt loams, sandy loams, what the original organic matter was. How do you think all that, I'm having struggle how you make some of the comparisons you're making across such diverse soils and such diverse climates and diverse cropping systems. I guess.
Well, I looked at each one on a site-by-site basis, and then only the effect of climate across the sites. The soils were silty loams, except for Prosser, which is more of a sandy soil.
In the native state, they started about the same organic matter, or they starting at different. I would have to think the drier areas would have much lower organic matter than the
Right,
wetter
yeah.
areas. How that might impact how these different practices.
Sure.
Because to me, the interesting part, if you had, they all run about the same way, is what are the interactions?
Right.
How does tillage work, not work in this environment versus this, to improve soil quality, cropping systems? My guess is that the environment has a huge impact on beginning soil characteristics that would
Absolutely.
influence on how all those things
Absolutely.
work. So that would be a very interesting